On this slide, we can see a drawing of the circular chromosome that we would find inside a bacterial or an archaeal cell. So in other words, this is the double helix of DNA that's inside the nucleoid. We've just removed all of the proteins that it gets wrapped up around in the nucleoid so that we're left with this circular double helix. And down here in the lower part of the drawing, they're showing us that the double-stranded molecule has been separated. The two strands have been pulled apart from each other. And remember, the very first place where that happens is called the point of origin, or what they've labeled as the origin of replication. Both of those terms are interchangeable. So we've pulled the two-stranded molecule apart to create two individual strands of DNA, shown in gray. Those are going to be the template strands. And on each one of those strands, we're going to start building new DNA. And the new DNA is drawn in red here. Now, as the DNA polymerase enzyme makes the new DNA, we're going to need to keep separating these strands. We're going to need to keep separating them over here and keep separating them over here. We're going to need the bubble to get larger in both directions. And that's what they're showing us over here. Look at how big the bubble has gotten now as we keep unwinding the double helix. So here's one old strand, here's the other old strand inside the replication bubble. And the enzyme is building new DNA onto the old DNA, shown in red. Now, here's another place where DNA replication gets a little bit confusing, and it goes back to those 5', 3' numbers. DNA polymerase can only build new DNA in one direction. It can only add new nucleotides onto the growing strand of new DNA at the 3' end. Okay, so we say that DNA is made from 5' to 3'. That is the only way that enzyme can work. So what that means is, inside the replication bubble, we're going to run into a little problem. If the enzyme begins making new DNA at the origin of replication, part of each new strand will be made seamlessly. It will be made continuously, just adding one new nucleotide after another, one after another, in one long continuous strand. But part of each new strand will have to be made very differently. It'll have to be made discontinuously, in other words, in fragments. And those fragments will have to be joined together eventually so that the new DNA is one long continuous strand. Now this sort of wrinkle in the process was discovered by a scientist named Okasaki, and so we refer to the fragments of DNA that are produced this way as Okasaki fragments. Now let's take a look at this weird process. What you see on this drawing is a piece of DNA that has been opened up in one area in a replication bubble. They've identified the origin of replication as being right here and right here. So the double-stranded molecule of DNA, the old DNA, was first broken open at the origin, and then the replication bubble got bigger and bigger and bigger as new DNA was built. The enzyme came up and started on this old strand and started making new DNA, shown in blue, in this direction, starting from the origin and heading off this way. And you can see that blue strand, that new DNA drawn in blue, is made as one continuous strand. The 3' end of the molecule is over here. So the enzyme just keeps adding bases to this end. The replication bubble keeps getting bigger and bigger, and you just keep adding a base, and then another base, and then another base, and then another base, all the way along the old DNA. We give this nice, smooth, continuous process of DNA replication a name. We call this leading strand synthesis. Now, the other term that's introduced on this slide, in this drawing, is the word fork. The place where the replication bubble sort of ends is referred to as the fork. So as the replication bubble gets bigger and bigger and bigger, the fork moves. In other words, the bubble opens up and it gets bigger, and the bubble keeps opening up and keeps opening up. So the fork, the place between the end of the bubble and the double-stranded DNA, the fork keeps moving. Leading strand synthesis heads into the fork, if you will. It just keeps going in a straight line. Over here on the opposite strand, remember the anti-parallel strand, the enzyme does the same thing. It comes in at the origin and it starts making new DNA. But because the opposite strand is anti-parallel, the enzyme is working in this direction. And the fork is heading in this direction. So the leading strand synthesis process is happening on both strands. It's just happening in different directions from the origin of replication. You with me so far? Here's where it gets weird. We also have to build DNA over here. You know, the enzyme comes in here and it just goes this way. Well, what about this side over here? You have to make DNA over here too. And you have to make DNA here. But the enzyme comes in and goes this way. So what does a cell do? Well, the enzyme can still work over here, but there's an issue because the enzyme can only make DNA in one direction. On this strand, it can only make it this way, towards the right. And on this strand, it can only make it this way, towards the left. So in these other regions, over here and over here, the enzyme is literally going to have to jump backwards and then make a piece of DNA. And then jump backwards and make a piece of DNA. And that's what they're showing us. So you have to use your imagination. But at one point, the replication bubble was just this big. It was just like right here. It was little when it first opened up. So the enzyme started here and made DNA in that one direction, this way. Then the bubble got a little bit bigger. Now the enzyme had to jump backwards, start here where the fork was, and start to make DNA in this direction. And then the bubble got opened a little more. So the enzyme had to jump back to the fork and make another piece of DNA in this direction. So in this area right here, and in this area right here, the enzyme makes new DNA in Okasaki fragments. And we refer to the process as lagging strand synthesis. So the take-home message is that on each of the old strands, part of the new DNA will get made using leading strand synthesis, and part of the new DNA will get made in Okasaki fragments using lagging strand synthesis. So let's summarize what we've said so far. Inside a microbial cell, a bacterial cell, or an archaeal cell, there's a circular double helix of DNA. And when the time comes to copy that DNA, that circle has to be opened up. The two strands of old DNA have to be separated from each other. There's an enzyme that is going to open up the double helix at the point of origin. We don't need to know the name of that enzyme. Just understand that there's an enzyme that does that. In order to begin copying each of the old strands, an RNA primer, a short little piece of RNA, has to be placed on each of the old strands. A different enzyme is going to do that work. That will help start the replication process. The DNA polymerase enzyme will come in and it will start making new DNA at the end of that primer. DNA polymerase will then build new DNA, but only in one direction, 5' to 3'. So that means part of each new strand will be made continuously. That's leading strand synthesis. And part of each new strand will be made in fragments, Okasaki fragments. And we call that lagging strand synthesis. Once the entire DNA is copied, so instead of having one double helix, now we're going to have two double helixes. Each one has one old strand and one new strand. Once that entire process is finished, the RNA primers will get removed and replaced with DNA. And yes, another enzyme does that work. And then finally, the Okasaki fragments will get joined together by yet another enzyme. The new DNA will be joined to each fragment and will have one long new strand inside each new double-stranded molecule of DNA. So this process is pretty amazing, and it happens so often. It happens every single time a bacterial cell or an archaeal cell, any microbial cell, divides. Every time a cell divides, it has to copy its DNA so that the two new cells, the daughter cells, can get a full and complete molecule of DNA. DNA polymerase is found in every single living cell on this planet. Now, bacterial DNA polymerase is a little bit different from human DNA polymerase, but the idea is the same. We copy our DNA in essentially the exact same process. The enzyme does this work very quickly. DNA polymerase works very quickly. And sometimes the enzyme makes mistakes. No biological system is perfect, and no enzyme is going to do its work perfectly. As DNA polymerase travels down the old DNA strand, the template strand, and uses it to build a new DNA strand. Remember, it has to match up bases in a complementary pattern. A's with T's, G's with C's. Sometimes the enzyme makes a mistake and it puts the wrong base. So it might match a T with a G or an A with a C. This has been studied to the point that we know how often DNA polymerase makes mistakes. Approximately one time out of every 100 to 1,000 bases that get placed will have a mistake occur. So that's pretty good for that enzyme. That enzyme is pretty efficient and pretty accurate. But about once every 100 to 1,000 times, the DNA polymerase enzyme will put the wrong base in the new DNA. Now, the good news for you and I is that cells proofread. They proofread their new DNA. And we've already talked about this in lab. Remember, we talked about the repair systems that are in place, and we looked at those repair systems in action after we exposed bacterial cells to ultraviolet light. We exposed the bacteria to UV light, and the surviving cells we then watched repair their DNA. Those were the cells that were able to grow colonies even after they had been exposed to ultraviolet light. So that's good news. That's good news for bacteria and for archaea and for eukarya, that our DNA has a system in place to fix mistakes. Proofreading systems, repair systems are in place. But there's a little bit of bad news too, because those repair systems are also biological systems, and they're not perfect either. So while most of the mistakes that the DNA polymerase enzyme makes get repaired, some of them don't. Some of them are left. Some of them persist. And those mistakes are what we call mutations. So all a mutation is a permanent change in the order of the bases in DNA. That's what a mutation is. Remember when we talked about ultraviolet light and the damage that it does? We said that ultraviolet light doesn't mutate our DNA. Ultraviolet light damages our DNA. Any place that there are thymines, T bases, side by side, the ultraviolet radiation can create what's called a thymine-thymine dimer. It can create this very abnormal bridge between those two bases. That's DNA damage. We said that sometimes that damage leads to mutation. And the time that occurs is during DNA replication, because the DNA polymerase enzyme comes along, traveling down the old strand, building a new strand, and it hits that thymine-thymine dimer, and it doesn't recognize it. It doesn't know what to put there. It doesn't see a T and then a T. It sees this weird structure called a thymine-thymine dimer, and it has no idea what base to put there. So it just puts any base in that location. The mutation is then created. So ultraviolet light damages DNA, and then when that DNA gets copied or replicated, it often creates a mutation in the process. So errors during DNA replication are one of the most common causes of mutations. Okay? Remember, even when DNA polymerase makes an error, most of those errors get corrected, but some of them persist. What else causes mutations? Well, as you all know, there are certain chemicals that living cells get exposed to that can mutate DNA. We call those mutagens or carcinogens, very dangerous chemicals. And finally, exposure to certain forms of radiation can also cause mutations in the DNA. If we go back to DNA replication and back to gene expression, and we think about the genetic code, inside the genetic code, as you recall, are groups of 3 bases, codons. And it's those codons that code for the order of the amino acids in a protein. When mutations occur, when bases are in the wrong order in the DNA, that changes the codon in that location. And if the codon is changed, at least if it's changed enough, we're going to get a different amino acid from that codon. And if we change the order of the amino acids, that's going to lead to a change in the protein. And if we change the protein, we're going to change the trait that that protein creates. So mutations are capable of changing traits in organisms. Now, I just want to state here that not all mutations are bad. We tend to associate mutations with disease, and yes, mutations can lead to disease, including very bad diseases like cancers. But it's important to remember that mutations are also the basis of evolution. Sometimes, when cells are mutated, the new traits are actually helpful to the organism. The new traits actually allow the organism to adapt better to its current environment. And those adaptations are what give organisms a survival advantage in certain situations. So while mutations are bad in some instances, we must remember that mutations can also be quite helpful to organisms, and that across long stretches of time, it's mutations that change organisms, that help populations evolve.